all the CUET aspirants. So, here we are in our first session of Pteridophyta, wherein from Pteridophytes what we are going to cover for our exam. Structure, reproduction and life cycle, of course, characteristic features of Pteridophytes, reproduction and life cycle. And then systematic position of Rhynia Lycopodium, Equisetum and Marsilia we are going to learn, after which stellar evolution, heterosporia and seed habit. These are the peculiar evolutionary features that we have to observe, because heterosporia and seed habit is the first condition which is observed for the first time in Pteridophytes. From here the heterospory condition is being driven to gymnosperms and angiosperms. More than type studies in Pteridophytes, most of the time you got questions on heterosporia and seed habit and on stellar evolution as well. Now, we will look into the general characteristics of Pteridophytes. First, see, Pteridophytes, primary vascular cryptogams. These are primary vascular cryptogams which produce spores and they lack seeds. Seeds are absent in Pteridophytes. If you see the origin, the term Pteridophyta, it comes from the Greek word pteron. Okay, it comes from the Greek word pteron, which means feather, and this refers to feather-like fronds. Okay, it refers to feather-like fronds. And Pteridophytes are addressed as first vascular cryptogams. They are first vascular cryptogams. Fine. Vascular cryptogams, these are seedless vascular plants which are invaded into the land and they reproduce via spores. Spores will act as sperm cells of Pteridophytes, and Pteridophytes are the connecting links between the bryophytes and seed forming spermatophytes, between non-seeded plants and seed forming spermatophytes. What are the non-seeded plants? Algae and bryo. Algae and bryophytes. And Pteridophytes, they are the connecting links for algae, bryophytes which are non-seeded plants, and seed-bearing spermatophytes. Seed-bearing plants are addressed as spermatophytes, both gymnosperms which produce naked seeds and angiosperms. Fine, right? And here if you see the life cycle has alternation of generations. The dominant phase in the life cycle is sporophyte. In bryophytes, if you see, the dominant phase in the life cycle is gametophyte, but here it is sporophyte. Sporophyte which is diploid and it is differentiated to stem, leaf and root. So, stem, leaf, roots are diploid here. Fine, right? And gametophyte is haploid, it is short-lived, but it is independent. In Pteridophytes, one of the major feature to be addressed is both the gametophyte and sporophyte, both are independent to each other. They are independent to each other. Fine. Cells will differentiate and they are well marked as sporophyte. Clear with the basic characteristic features and alternation of generations, both gametophyte and sporophyte of Pteridophytes. Now, if you see the habitat of Pteridophytes, they grow in cool, moist and shady places. Some of them will also act as xerophytes. For example, Selaginella rupestris. Okay, this is a xerophyte which inhibit the high temperature areas. The adult plant body is of course a sporophyte, resembling same like that of flower plant, but flower is not the reproductive organ of Pteridophytes. Okay. So, the dominant plant body is sporophyte. In that case, root, stem, leaf will be diploid in Pteridophytes. Sporophyte is independent of gametophyte, and here if you see the stele, okay, stele contains xylem and phloem, and they may be either protostele or siphonostele. Two types of steles are formed, either protostele or siphonostele. The primitive form of stellar arrangement is observed in Pteridophytes. Next, what will be the leaf structures and reproduction? Leaves are addressed as sporophylls in Pteridophytes. sporophytes, okay. They are sporophylls because they bear spores. They are spore bearing. Leaf structures are spore bearing. Spore bearing. So, here leaves will be small, minute and sessile in Lycopodium. They are petiolate and megaphyllous. They are petiolate and megaphyllous in Felicinae, okay. And reproduction is via spores. Spores are produced in the sporal sacs or spore sac, which is present over the leaf. The spore sac is known as sporangia. Inside sporangia, spores will be formed and they will be dispersed out. As they are dispersing out from the leaf structures, these leaves are addressed as what, ma? Sporophylls. Important point to be noted. And here, look at the sporophyte type. As I told you, heterosporous condition is first observed in pteridophytes. Here you see both homosporous condition and heterosporous condition. Homosporous condition where all spores are similar in shape and size, whereas heterosporous condition, the large spores are known as megaspores. Okay, these are megaspores which give rise to, megaspores will give rise to female gametophyte. Female gametophyte. And small spores which are formed here, small spores, they are addressed as microspores. Microspores. These microspores will eventually develop into the male gametophyte. Fine. And here you see sporangial development. Two types of development will happen if the sporangia is differentiated from group of superficial cells. If sporangia is developed from more than one cells or group of superficial cells, such type of sporangiate is known as eusporangiate. But if sporangia is developed from a single initial cell, that is known as leptosporangiate type. Okay. Eusporangiate best example is Selaginella, leptosporangiate example is Marsilia. Examples are always important, I am telling you, examples are always important because from examples only the questions will be picked and asked. Fine, right. Next, gametophyte in pteridophytes is prothallus. Prothallus is the primary gametophytic structure. Primary gametophytic structure in pteridophytes. Primary gametophytic structure in pteridophytes is prothallus. This is developed from haploid spores. Okay, this is developed from haploid spores. Prothallus is independent and it is autotrophic due to chlorophyll. Clear till here? And this prothallus will later mature to a developed sporophytic plant. Next. Prothallus, what does it contain? It contains both male and female sex organs. Here also you can see both monoecious and dioecious conditions. And water is essential for fertilization, right? Both for bryophytes and pteridophytes, water is essential for fertilization, and post fertilization this will form a diploid zygote. Diploid zygote is formed post fertilization. Next, zygote will then develop into a sporophyte. Embryo development, if you see embryo development, it shows clear morphogenesis into leaf, stem and root. Clear morphogenesis is being shown. Next, alternation of generations is vital feature for their life cycle. Clear? Next, Rhynia is the first type study we are going to learn. Today we are going to learn two structures. One is Rhynia and second type study we are going to learn is Lycopodium. Focus and listen. First, look into the schematic position of Rhynia. Genus Rhynia, family Rhynaceae, order Rhyniales, class Coelophytopsida, division Coelophyta. This is the schematic position. Look into the history and discovery. This is discovered by Kidston and Lang. Okay, discovered by Kidston and Lang from Rhyniate bed. Okay, this is in which era? Middle Devonian era. Species you see Rhynia guanea and Rhynia major. These are the two species of this plant. They grew alongside Horneophyton and Asteroxylon. They grow along with this. This is thought to be from which era? Middle Devonian period to now it is believed to be in lower Devonian as well. Okay, this is the history of this plant. Now, external features of Rhynia guanea. Okay, so here we are going to learn two species. Yeah. Look into the comparison of both the species directly. Here we see first stem. Stem is around 200 meters to 2 meters in diameter. This will not be asked. Okay, stem height will be very less. Stem will either have hemispherical outgrowths. These are because of rudimentary leaf-like structures over the stem. Whereas in Rhynia major, the stem will be smooth. And here in Guanea you can see flask-shaped archegonia. There is no such structure in Rhynia major. Adventitious branches are formed, and here aerial shoots are free from such branches. You can see they are reproduced asexually, vegetatively and sexually. Whereas Rhynia major, this is reproduced only asexually. Sporangium is of this height and diameter, and here the diameter of sporangium and height is more when compared to Rhynia guanea. Spore diameter is 4 microns, here the spore diameter is 65 microns. Clear? This is the clear differentiation between two species of Rhynia, morphological differentiation. And here we go. With the internal structure and anatomy. Look into the general structure. Epidermis is overcoated by thick cuticle which is interrupted by stomata. Okay, epidermis is interrupted by stomata in the stem. Cortex vascular tissue is also present. Epidermis which has cuticle layer over it on which stomata is present, and inside which cortex region it is not further well differentiated. Cortex is just differentiated into two layers in Rhynia, outer cortex and inner cortex. Outer cortex is larger than inner cortex and it has distinct intercellular spaces. It is connected to stomata. Inner cortex will contain chlorophyll which is of photosynthetic activity. So, look at here the anatomy. Epidermis encoded by cuticle. This cuticle is interrupted by stomata. Fine, right? And below which you have two layers, which is outer cortex, then inner cortex. Outer cortex and inner cortex. Outer cortex is larger and it has distinct intercellular spaces. It is connected to stomata. Inner cortex, they contain chlorophyll which is of photosynthetic activity. The cells here are of photosynthetic activity. Next, vascular tissue you observe. Central vascular strand is very simple in organization. How xylem is present? Xylem is just made up of tracheids. That is the reason you observe pteridophytes, gymnosperms and angiosperms are addressed as tracheophytes. What are the first tracheophytes? First tracheophyte land plants are pteridophytes. Pteridophytes are also addressed as tracheophytes. Xylem is made up of tracheids. It has annular and spiral thickenings. Okay. Central tracheids will be smaller than the peripheral ones, and this indicates endark xylem. Endark xylem which is differentiated into protoxylem at the center. What is endarxylem? If protoxylem is towards the center and metaxylem is towards the peripheral end. Fine, right? Protoxylem is towards the center and metaxylem is towards the periphery. That is the arrangement of xylem. Next, phloem. How is phloem arranged in Rhynia? This is made up of thin walled cells. It does not have any intercellular spaces. There are no sieve plates. Just primitive phloem is present. Unlike modern pteridophytes, in Rhynia it just has some primitive phloem. What are the observations by different scientists? This is not required in the type study. Yeah, internal structure of phloem you see. Presence of sieve tubes and parenchymatous cells are observed, and sieve tube cells will have pores as well. These pores have lateral cell walls. Pores are found in the lateral cell walls, and pit fields you can see, which are inclined pit fields. There is no pericycle and endodermis. Cortex is not further, cortex or stellar region is not further differentiated into pericycle and endodermis. The structure, internal anatomy of Rhynia is very simple. Epidermis, above which cuticle is present, which will be interrupted by cuticle. Below the epidermis, if you can see cortex region, two layers of cortex regions are observed, outer cortex and inner cortex, and inside which you have vascular tissues, both xylem and phloem. Xylem just has tracheate cells, phloem will just have some sieve pores and primitive phloem structures, but there are no sieve plates observed. Next, rhizome anatomy. What you see in rhizome, which is root like structure, this is similar to that of aerial stem, but it lacks clear distinction or differentiation between outer and inner cortex, whereas in stem anatomy you can see clear differentiation between outer and inner cortex. In rhizome of Rhynia it does not have any clear differentiation between the outer and inner cortex. Xylem is very minimal, only few tracheates are present. Fine, right? Primitive xylem or very minimal xylem with few tracheates is observed. Next, apical region will contain numerous dividing cells and they have no distinct apical cell, Sporangium, this is very important. What will be the shape of sporangium in Rhynia? Club shaped or cylindrical shape, as there are no leaf structures in Rhynia, which is primitive pteridophyte. The sporangium is observed at tips of aerial stems. They are observed at the tips of aerial stem. Sporangium will be having three layers. Outer one is thick, outer layer is thick and it has cutinized epidermis. Middle layer is thick walled and inner layer will have delicate inner layer is present. Outer cutinized epidermis, middle thick walled and inner will have delicate inner wall layers are present. Next, spores. Numerous spores are arranged and spores are always arranged in tetrad condition. 4 spores will be having. And Rhynia will show homosporous condition only, same all spores will be of same type, and spores are trilate with 3 rayed mark. Spores will have a 3 rayed mark like this. Fine. And then what type of sporangium? Initially we have learned 2 types of sporangium, eusporangium and leptosporangium. Sporangium is formed from group of initial cells. So here in Rhynia the sporangium type is eusporangium type. Very important ma. How cells are divided to form? Cells will divide to form outer primary cell and inner spore producing cell. And gametophyte structure we have already learnt how gametophyte structure is. Then primitive characters of Rhynia. What are the primitive characters? Dichotomous branching of the stem, major character to be highlighted. There is no root, only rhizoids are present or rhizome structure is seen, and there are no true leaves. That is why sporangia is present at the tip of the aerial stem. Photosynthesis occurs in green aerial stem only. Vascular bundles is haplostele. Vascular bundle is haplostele, where the central stand of the xylem is observed. Okay. haplostele, which is present in the central strand of the xylem. Sporangia is formed at the terminal tips of the aerial branch. Next, roots. Primary roots are present of the young sporophyte, which is ephemeral, short-lived. Primary root is short-lived, okay, and it dies early. Adventitious shoot they will develop later, and here roots also show dichotomous branching. Stem shows dichotomous branching and roots also show dichotomous branching arising endogenously. This arises endogenously from lower side of the prostrate stem. In erect stem, roots arise from the basal part of the stem. Okay, roots will arise from basal part of the stem. Fine. These are called cortical roots. Roots which arise from the basal part of the stem are addressed as cortical roots. They do possess root cap and root hairs occurs in pairs. Root hairs will occur in pairs. So, what are the characteristic features from the gametophyte if you observe? Dichotomous branching of the root, and roots grow endogenously, haplostele, and roots which grow at the base of the stem known as what? Cortical roots here, and roots do possess root caps and it has a pair of root hairs. Next, if you see stem, of course dichotomous branching, this branching can be equal or unequal branching is observed. Next, leaf, this is important, observe, yeah, leaf if you see opposite phyllotaxy is observed, opposite phyllotaxy, just a second ma. So, after this primitive characteristic features of Rhynia, from root structure which we are learning is from Lycopodium. Root structure in Lycopodium, it has dichotomous branching, endogenously formed roots, cortical roots, root caps with root pair of root hairs. These four are the characteristic features, followed by stem and then leaf. Leaf will possess opposite phyllotaxy. Leaves will have single median wheel, one single median wheel. This will not reach the tip, and this is addressed as what? Microphylls, because it will possess only microsporangia. And leaf margin, it can be serrate leaf margin and decorate leaf base is present, and this exhibit anisophilous condition. What is anisophilous? Leaves will be of different shapes or sizes, which is anisophilous condition. Fine, right? This is about the leaf structure. Next, TS of stem, important one. TS of stem, if you can see, it has epidermis, yeah, here we go. It has epidermis. Epidermis is made up of parenchymatous cells, and it possesses cutinized wall layer above the epidermis, and it also has stomata. Below the epidermis, the cortex is divided into three regions. Outer cortex is known as hypodermis, middle cortex is parenchymatous cortex, inner cortex will be sclerenchymatous cortex. Okay. Outer cortex is hypodermis, middle cortex will have thin walled cells which are parenchymatous cells, inner cortex is endodermis which is made up of sclerenchymatous tissue. Next, comparison between different species is not required for you. Pericycle. Okay. Endodermis is absent. Yeah. Endodermis and pericycle. Endodermis will mark as the last layer, sorry, and from pericycle this marks what? Stellar region. Above pericycle till endodermis it is extrastellar region. From pericycle it will be stellar region. Pericycle, protoxylem, metaxylem and pith are present, you see. Pericycle is just located inner to the endodermis. This is composed of one or more layers of parenchymatous cells. One or more layer of parenchymatous cells. And xylem region, if you see, this is present in compact central zone only. Okay, this is composed of primary xylem and primary phloem itself. Stele type, what type of steles are seen? They have central protostele, and if you can see actinostele which has star shaped xylem, radiating arms. Xylem will have radiating arms like this. Here you can see star shaped xylem. Xylem with radiating arms. And a mature plant with complex variations, this occurs in different species. Xylem is always at the center and this xylem is surrounded by phloem tissues. Xylem surrounded by phloem tissues. Clear, right? What type of stele is seen? Primitive protostele or actinostele is observed in Lycopodium. Here you observe actinostele first. Actinostele, it is star shaped xylem and arrangement where xylem which is present at the center, it will radiate out its arms. Observed in different species. Here you have to remember the species. Different stellar arrangements in different Lycopodium species. Here you can see this in Lycopodium serratum, Lycopodium phlegmalia and Lycopodium silago. And this you observe, plectostele, where xylem is arranged in plate-like structures. Xylem arranged in plate-like structures, and phloem is found in between the plates. Phloem is observed in between the plates. Xylem is present at the, is arranged like plates, and phloem is in between the xylem plates like this. Okay. This is plectostele, the name itself is same. Examples Lycopodium volubile and Lycopodium clavatum, followed by mixed protostele. Structure, xylem and phloem are uniformly distributed in mixed protostele. Xylem is found in scattered strands or irregular tracheids are found. Scattered strands or irregular tracheids. Examples you see Lycopodium cernum, Lycopodium lateralis, Lycopodium drummondi. Okay, these are the examples of what? Mixed protostele. These are the different types of stellar arrangement that you see in the stem of different species of Lycopodium. Actinostele, plectostele and mixed protostele. Next followed by TS in Lycopodium. Okay, TS, transverse section of leaf in Lycopodium. Here if you see epidermis is single layered epidermis is present with stomata at the basal regions of the epidermis, and it has thick cuticle, they contain stomata. Most of the species of Lycopodium you can see stomata on both the surfaces, which is addressed as isophilous type. Isophilous type where stomata is equally distributed on two surfaces of the epidermis. But in few species the stomata is anisophilous, like if you see in Lycopodium volubile and Lycopodium complanatum, the stomata is anisophilous. Fine, isophilous and anisophilous stomata. Next, mesophil structure. Mesophil is undifferentiated tissue, it has vascular bundles. This contains single unbranched vascular strand. Single unbranched vascular strand, this traverses the leaf, and this is of what type? Mesarc type. Protoxylem and metaxylem are not well differentiated in Lycopodium. Phloem and xylem you see here. Phloem is present at the peripheral end, it will be surrounding the xylem tissue, whereas xylem is present at the center. Okay. Tracheids, of course, they are tracheophytes. Pteridophytes are tracheophytes. Tracheids are of different types, like spiral or annular thickenings are present. Phloem is composed of sieve tubes and phloem parenchyma. Unlike Rhynia, which is very primitive, it does not have any phloem structures, but Lycopodium has what? Sieve tubes and phloem parenchyma. This is to be noted. Next, endodermis is not always distinct. Root, if you see, we have observed stem and leaf. Now, root structure. Epidermis, it has numerous root hairs. Okay, root hairs are formed by anticlinal division. Next, cortex, unlike stem region where it is divided into 3 layers, here in the root it is just outer cortex and inner cortex. Okay, outer cortex is composed of thick walled cells which are sclerenchymatous. And then inner cortex is parenchymatous, and last region of the cortex is endodermis. Vascular tissue is well differentiated into metaxylem and protoxylem, which is surrounded by a sclerenchymatous sheath. Both xylem and phloem tissues or vascular strands are surrounded by a sheath, okay, that is sclerenchymatous sheath. Next, reproduction in Lycopodium. What type of reproduction is taking place? Vegetative reproduction, asexual reproduction and sexual reproduction. Vegetative reproduction in Lycopodium is sporophyte, and this is capable of vegetative propagation. Fine, right? Vegetative structures, if you observe, gymma or bulbils, developed at shoot apices. At shoot apices, gymma or bulbils are developed. In the place of leaves, gymma or bulbils are developed at the shoot apices. This is found in different species like Lycopodium silago, Lycopodium phlegmaria and Lycopodium lucidulum. The structure is reduced axis which is surrounded by thick and fleshy leaves. Functions of bulbils, which are the storage regions which stores food, and when gymma will fall off from the parent plant, this will give rise to young plants again. Next, formation of resting buds, they are found at the apical buds of the rhizome. Where are the resting buds formed? They are present at the apical buds of the rhizome, and here stored food, it will become bulged with crowded leaves. This is seen in Lycopodium inundatum, and these are called as resting buds. Different types again. Fragmentation is most common mode of asexual reproduction. Formation of root tuber cells, okay, where parenchymatous region of root cortex will form tuberculous. These cells will store food, they have thick walls and they can germinate when they land onto the suitable substratum. Next, formation of adventitious buds. So, these are found in basal leaves near the ground. They are formed due to epidermal cell proliferation, and they are also induced by decapitation of stem axis, observed in Lycopodium silago. Asexual reproduction and sporogonium you see. Asexual reproduction, all the methods we have observed, and here you observe in Lycopodium there are many sporophylls which are formed at the tip, which are addressed as strobili or cones. Here we go with structure of sporangium. Sporangium which is present in strobili or cones. Yeah, see here, sporangium will have two parts. Basal stalk is present and upper capsule is present. Capsule is unilocular, which means it is having single chamber and it is not having any lobes. This is surrounded by 2 to 3 layered walls. It is surrounded by 2 to 3 layered walls. It contains spore mother cells. It contains spore mother cells. Okay. Tapetum, which is the innermost wall of the sporangium. Okay. Tapetum is the innermost wall of the sporangium. It has the nutritive layer, it will nourish and support the spore development. Next, it is composed of granular cytoplasm and it has large nuclei. This will help to round the spore mother cells. Clear till here? Tapetum will provide nourishment to the spore mother cells. You see the sporangial wall and tapetum. Tapetum is the innermost wall which will provide the nutrition. It will stay intact, but it will not disorganize. Archisporial cell will undergo further divisions forming out the spores, inside which you can see sporogenous tissue which is having separate cells inside it. Each cell will distinguish or develop into a spore mother cell or a sporocyte. The spore mother cell undergo reductional division inside the capsule itself, reductional division or meiosis inside the capsule itself, and it will form 4 haploid spores. Fine. Mature sporangium will be in what shape? Kidney shaped with small stalk. Yeah, how the spores will get dehiscence? Okay, from the sporophyll head, internodes will stretch and sporophylls will spread. They dehiscence through thin lining or weakening the stomium region. Okay, through stomium region weakening the sporangium wall, the spores will be dispersed. Where do you see stomium over the sporangial wall? This is small line of cells which are present over the upper surface of the kidney shaped sporangium. This is hygroscopic in nature, loses water, dehydrates the wall so that spores will be liberated out easily. Yeah, gametophyte we have discussed already. So now if you see the life cycle, yeah, if you see the life cycle carefully, observe this. Sporophyte stage, first Lycopodium sporophyte, because sporophyte is the main plant body. Which is diploid, right? This will have strobili or cones at the tip, which is diploid. It consists of sporophylls, they have sporangia and each different separate cells. They will turn to form a sporocyte or spore mother cell. Sporocyte or spore mother cell is one and the same. This will undergo reduction division forming out spores. From here the gametophytic stage will start. The spore on germination will first develop into a primary gametophytic structure known as prothallus. Prothallus will further differentiate and it will develop into a mature leafy gametophyte, after which it will bear the sex organs, both male gametophyte and female gametophyte. Antheridium will be releasing out spermatozoids or sperm cells or antherozoids, anything you can call. Sperm cells or antherozoids. And archegonium, which is haploid, it contains egg cell inside it. Fertilization. Spermatozoid when fuses along with the egg cell, this post fertilization forms a diploid zygote, which is also addressed as oospore, which is diploid. This will undergo mitotic divisions, mitosis, and develops into an embryo. This develops into a new sporophyte. Clear till here? Any doubt anywhere? This is the complete life cycle of Lycopodium. Fine? Any doubts anywhere, ma? Here you see once again that it has equal gametophytic and sporophytic phases, of which is the dominant phase? Sporophytic phase will be dominant because the main plant body is sporophyte. Now life cycle is clear to us. Once exercise the ploidy as well. As the main plant body is sporophyte, remember the root, stem and leaf structures along with strobili, everything will be diploid itself. And what do you address leaf structures as? Microphylls, because they are smaller in structure. And sporophyll, spore mother cell, still here all the ploidy will be diploid. From spore, prothallus and the gametophytic structures will be haploid. And then post fusion or post fertilization, again you can see the diploid structures. Okay? So this will be the complete life cycle of Lycopodium. Once if we take the keynotes of Pteridophytes, what we have completed today. Yeah, from this structure, reproduction and life cycle of Pteridophytes is clear. We are done with Rhynia and Lycopodium. In our next class, we will look into Equisetum, Marsilia, Equisetum and stellar evolution. One more class required for heterospory and seed habit will be done. Okay. Once the complete pteridophytes are done, then we will move to gymnosperms. 2 massive sessions I will be taking on the objective of complete Archegoniate, which includes Bryo, Pterido and Gymno. For those 2 objective sessions, whoever are attempting for CUET Botany PG entrance, if you are attending that, that will be a biggest help for you, because around 100 to 150 objectives of Archegoniate I will be covering it. That will be useful for everyone who is attempting. Okay. So, that is all for today's class. I will see you in next class with the topics Equisetum, Marsilia and stellar evolution.